Hi everyone, welcome to the first chapter of Biology 1406 class for this semester. If you have watched my introduction video, you should know that my name is Dr. Mac, okay, and I am your professor for this online 1406 class. Now, before you watch this particular chapter 1 video, I hope you have gone through the class introduction video to know all the ins and outs of how this class will be. You know, where to access materials, how to stay on track with due dates. It is very important that you have gone through those instructions, or else you will feel very lost in this online class. Now, as I have mentioned in the class introduction video, you should have a copy of the lecture slides in front of you to follow along, whether it be printed or electronic version, and you should be actively taking notes and highlighting key terms to help you stay engaged during the lecture video. If you're just sitting there listening, it can get very boring and you might even fall asleep. So make sure, try to find a way to keep yourself engaged and so that you have some sort of interaction as you're going through my lecture videos. So let's have your notebook out and a copy of the PowerPoint slides out for chapter 1, and we will get started. Now, chapter 1 is basically an introduction chapter that sets the foundation of what biology is, how the information is discovered, and what different branches of biology there are. Now, biology itself is defined as the study of living organisms and their interactions with one another and their environment, okay, all the way from microscopic level to macroscopic level. What do we mean by those? Well, microscopic level is where organisms cannot be seen with the naked eye, but with the help of a microscope, which is why your lab 2 teaches you how to use a microscope, because a lot of the things we see and study in biology cannot be seen with our eyes alone. There are tiny, tiny little creatures that we will study in this class and learn about in this class that require the use of microscope. And also for your upper level biology classes such as microbiology and anatomy and physiology, where we're looking at microscopic tissues and cells. So, for example, right here, we have a picture of E. coli bacteria, a bunch of them, not just one. No, every little one of these is a E. coli bacteria. And, you know, there are normal versions of E. coli bacteria that live in our digestive tract to help with the nutrient absorption in the body. You know, but there are also virulent versions of E. coli, as you have all heard of before, that can lead to disease outbreaks like when E. coli was found in spinach and resulting in a lot of people getting sick. And that's why there will be news articles out there that says don't eat spinach for now because we have discovered that there is E. coli contamination in this particular branch of spinach. So you might think, oh, well, you know, I thought E. coli was those bacteria that normally live in our gut, but you know, there are other strains of bacteria out there, and how they interact with their macroscopic organisms around them can be different. Now, the second picture here shows these rock-looking structures where they are formed by layering of what's called cyanobacteria in shallow waters. Years and years and years ago, of course, it takes a long time for all those tiny little creatures to become a big, huge rock that we see, on the beach or something. And hence, both of these give examples of how microscopic organisms and macroscopic things in the environment always have interaction with each other and result in an impact to the surroundings in which we live in. Now, we must understand the basic biology in order to find cures to many human diseases, such as, for example, cancer. Now, even though we don't have a permanent cure for AIDS yet, but there have been medications that was discovered to slow down their growth. And that's why there's still some sort of treatment available when a person discovers that they do have AIDS. Okay, we can slow down the process with some drugs that specifically target, how fast the virus is able to replicate inside the individual. Now, there's a branch of biology called ecology that study our environment and how it is impacted by our human behaviors. Without really understanding those relationships, we would not be able to come up with ways to protect the stable ecosystem of our planet, which is, a whole new other branch of biology that we won't talk about in this class, but you may hit that particular topic when you get to biology 1407 if that is a requirement for your major. But ecosystem talks about how different interactions between organisms, such as populations, is affected by human behavior, such as breaking down of trees and forests and building of human structures that takes away animals' homes and hence affecting their population. And this is where we talk about animal extinction or endangered species, etc. Okay. And I won't dive too deep into that since we won't talk about that in this class, but just a brief introduction. Now, I'm sure most of you have heard of what the scientific method is. We always begin many biology courses by talking about the scientific method. It was first defined by a man in this particular picture here, named Sir Francis Bacon in the 1600s. Now, the scientific method involves research that uses defined steps, including observations and experiments. And these are very important things as being part of that protocol. It usually begins with an observation of some sort. I didn't really list observation here as one step, but if we were to break down this a little further, I would add observation being first before we can formulate a hypothesis. So we begin with an observation of some sort where one sees something happening out of the ordinary. Then one can then question, well, why did that just happen? How did that happen? Is there some reason to that or something caused that to happen? Get a whole discovery of gravity when an apple was, seen drop from the tree. Something very simple in life that we don't really ponder about, as a normal person, but somebody will come up with ways to explain things. Okay, so with an observation, then one will come up with a possible explanation for the observation in form of a hypothesis. And a hypothesis should really be something or statement that is testable and falsifiable. Okay, you will learn about these two characteristics of a hypothesis in lab as well. But these are two criteria that a well-formulated hypothesis should have. It should be testable through, experiments and whatever tests you come up with, and it should be falsifiable. Whether it is true or false, there should be a way to tell. Okay, then one has to come up with an experiment. And this experiment is used to test a hypothesis, which is defined as a series of repeated tests, experiments. That's what experiments are. They're repeated tests that will either support or falsify your formulated original hypothesis. Then comes the results. These are results you get from the experiments themselves, which will collectively, act as a summary of data, of information we can interpret as supporting or rejecting your original hypothesis. Now, if the data agrees with your hypothesis, then it supports it. And if your data disagrees with the hypothesis, then it rejects it. So it's that simple. Okay, it's just whether your results will confirm that that your hypothesis was correct, okay, or false. Now, here's an example of how the scientific method can be applied. A very simple example just to get you started. Now, we first begin with an observation. Okay, so for example, in this particular observation here, we're looking at how drinking coffee wakes me up in the morning. Okay, as many of you also experience, unless you're tolerant to coffee, of course. So a possible hypothesis that we can come up with is that maybe coffee contains substances that stimulates the brain such that I don't feel tired. Okay, so that's a plausible hypothesis where it includes things that I can test and also falsify. So the experiment we can design to test this hypothesis is simply to try to drink coffee at night and see if it wakes me up the same as in the morning. Okay, so very simple experiment just to see, well, is it time limited? If it's not time limited, there's got to be some stuff in there that wakes me up regardless what time of the day I drink the stuff. And then at the end, you have the result, which we will get either accept or falsify your hypothesis according to the results from the experiment. And of course, we know coffee has caffeine in it, so that, and we know that it will do what it's supposed to do regardless morning or night. Okay, of course, like I said, unless you're tolerant to coffee, and then that's a different story. But we're talking about a normal, natural person that is, does get affected by caffeine. So in the end, this will accept our original hypothesis that there is something in coffee that we know as caffeine that will wake us up and stimulate our brain. Okay. So this type of scientific method model is not only used by biologists, but also almost all fields of study can apply it as a logical, rational, problem-solving method. Okay, you always go from observation of some sort, formulating a hypothesis, making and designing experiments, and then you will collect results to either confirm your hypothesis or reject it. And then if it's rejection of a hypothesis, you just come back right up here and start all over again with another newly formulated hypothesis. And this can be applied to physics, to any daily problem solving, computer science, anything. It doesn't have to be biology specifically. Anything in life when we're trying to solve a problem, we're really going through with this scientific method unknowingly. Okay, for example, if your car breaks down all of a sudden, it doesn't start, well, that's an observation. So you come up with a possible hypothesis that something else is going on with your car that you can't start it. Okay, etc. etc. Okay, so another important thing to know about science is that it is something that can always be modified or changed if new discoveries were able to support the change given enough amount of data and evidence. So don't think that science is forever a fact. Okay, whatever it claims in a textbook, whatever it claims in any publications out there, it's not going to be, nothing is permanent fact. If later on we prove that something else says that is not true, we can always modify scientific information. Kind of like how a long time ago people used to think the earth was flat. Okay, now we know that the earth is actually round and spherical based on what we can observe through satellites and, being able to travel to outer space and see it with our own eyes. So, and another example that we will talk about when we get to the DNA chapter towards the end of the semester is that scientists used to think that proteins was the genetic heredity material. Now we all know that it is DNA that was proven to be the genetic material. We'll be in trouble if proteins was the genetic material because proteins are so affected by different conditions such as differences in temperature, low temperature, high temperature, etc. But DNA proved to be a very strong molecule that can hold our genetic material in a stable manner. And we will talk about a few of those experiments that was able to provide enough evidence to change the original thought that protein was the genetic material when we get to chapter 14 to talk about DNA. So now that we understand what a hypothesis is, the next step we need to do is to design an experiment to test our hypothesis. So what makes up an experiment? Now, an experiment always includes what's called a variable. And a variable pretty much is any part of the experiment that can be changed, okay? That's why it's called a variable, because it can be varied in a sense, okay? So for example, in this particular experiment right here, they are testing a population of individuals over the age of 65 for the effects of caffeine on memory. So in a well-designed experiment, usually we can only change one variable at a time, so the conclusions will not be obscured due to changing two things at the same time. For example, if you change two things, okay, A and B, and then you get a certain results. But how do you know if that result was due to changing A or was it due to changing B? Because you change two things at the same time. You can't distinguish the two. So that's why usually in every well-designed experiment, only one variable is changed at a time. Okay, and you will see this when you get to lab 5 this semester. When we observe different effects of liquids such as alcohol or caffeine on the heart rates of little creatures called Daphnia. Okay, so that's one variable we're changing. We're not changing anything else. We're keeping everything else the same so that our conclusion can mean something. Okay, so in this case here, okay, right here in this particular experiment, okay, what is the variable between the two groups? What do you think it is? Yep, it is the caffeine, okay? That is the variable that's being changed between the two population of individual. Everything between the two groups are the same. The only thing they change between them is this experiment was one that was given. One group was given decaffeinated coffee, while the other group was given caffeinated coffee to see the effects of caffeine have on memory. In terms of sex, they're made up of both men and women. In terms of population size, they're both made up of 40 individuals. In terms of age, they are all over the age of 65. So these are some of the things that are kept the same, and the only thing different between the two groups here is the caffeine. Okay? All right, and hence the group that did not get the variable that's being tested is called the control group. And you will have to learn about this when we get to lab 5 and identify what's the variable, what's the control, etc. Okay, so control group is the group within the experiment that is not given the change. Okay, they have everything as the same. It does not contain that one thing we're changing, while the group that is given the variable is called the experimental group. Okay, and in this case, the control group is the decaffeinated group, okay, where they didn't receive caffeine just like any other normal individuals, while the experimental group is the group that receives caffeine, which is our main variable in this particular experiment. Now, the control group usually contains every feature of the experimental group other than the variable, which was what I was talking about in terms of age, sex, population size, etc. So if the data that was collected from the experiment were inconsistent with the original hypothesis, then the hypothesis is rejected. Okay, and then that doesn't mean it's a bad story. Okay, we don't throw that data away because it's valuable information. That just means that hypothesis was wrong. So now we can formulate another hypothesis, and we can propose another one and design another experiment to test that next hypothesis until we get it right. Okay. All right. So here's a quick practice for you to identify the components and steps of the scientific method to this particular example. Let's go through these 4 statements and see if you can identify which is the observation, hypothesis, experiment, or result. Okay, let's go through these. In statement A, it says there is something wrong with the electrical outlet. B, my toaster doesn't toast my bread. C, I plug my coffee maker into the outlet. And D, my coffee maker works. So, which of these statements do you think is an observation, hypothesis, experiment, or result? Feel free to pause the video. Try to test yourselves, okay, before we reveal the answers. So take a pause. Okay, so first of all, the observation, which is the first thing you do, is you notice that my toaster doesn't toast my bread. Okay, so this is what I see happening. I put my toast in my toaster and it doesn't toast. Just like how I was talking about, I get in my car and it doesn't start. That's an observation. So you come up with a hypothesis. And the hypothesis is, in this case, there must be something wrong with the electrical outlet. Okay, so that's somehow this particular hypothesis. And of course, another hypothesis could be there is something wrong with the toaster. The toaster is broken. Okay, but in this particular case, the hypothesis that was given was that there is something wrong with the electrical outlet. So the experiment is, well, let me plug something into the electrical outlet, then I can figure that out. Because my hypothesis was the electrical outlet is the problem. So in order to test my quote-unquote electrical outlet, then you grab another appliance and plug it in and see if it works. So that is your experiment. Plug in something else, which is the coffee maker in this case. And the result that you observe is that my coffee maker works when it is plugged into the electrical outlet. So that confirms that actually falsifies your original hypothesis, because in your original hypothesis, you think that something is wrong with the electrical outlet, but when you plug in the coffee maker, it works. So that means there is nothing wrong with your electrical outlet. Okay, so this result right here rejects the original hypothesis. Okay, and hence that's not the end of the story. That doesn't mean it's something, oh my goodness, I just wasted my time. No, you have just gotten rid of one possible thing that can go wrong. So now the next hypothesis that one most likely would come up with then is there is something wrong with my toaster. Okay, it is my toaster that's broken. Okay, then you of course you design another experiment to figure what's wrong with your toaster. Okay, all right, so that's just a quick practice in identifying the different parts of the scientific method. All right. Okay, so now that we have what we have just discussed is really just a simplified version of the scientific method. Here are the detailed steps that include some of the things we didn't talk about. We did talk about observation, but usually after the observation, you kind of did this in your head, but if we want to write out everything, we really should write out all that stuff that our thought process go through. Usually after I make an observation, you then ask a question. You would say, oh, why did that happen? Okay, or go back to the broken, my toaster didn't work story. Then you would think, oh, then what is wrong with my toaster? Or is there something wrong with my electrical outlet? So that's a question that you form before you can come up with a hypothesis. So after you come up with a question, then you form a hypothesis that aim to answer that particular question. Okay, in that particular story case, then you would think, okay, is it the electrical outlet's fault that has caused my toaster to not work properly? So here's a hypothesis that I have for this problem. Now, another thing that we didn't talk about is usually we would make a prediction based on your hypothesis. This is something you will have to do for your lab 6 when you write your lab report. I'll talk a little bit about your lab report when we get to the different parts of a scientific paper in a bit. But usually after you formulate your hypothesis, you would make a prediction, okay? Thinking that if we go back to that same story of a toaster, then you would say, okay, if it is my electrical outlet that doesn't work, something's wrong with my electrical outlet, when I plug this coffee maker in, my coffee maker should also not work, okay? So that is a prediction. Then once you have your prediction, then you would do your experiment and test that particular prediction. And after you get your results, you would analyze your results just as we talk about. And then you would either say that your hypothesis was correct or hypothesis was incorrect. And then you would report your results. If, whether it's correct or incorrect, you still report it. But if it is incorrect, then you go back to this step and form another hypothesis like we just did. Well, the electrical outlet hypothesis was wrong. It was rejected because my coffee maker works. So then you formulate another hypothesis that something is wrong with your toaster. Okay? All right, so even if an experiment did not necessarily agree with your original hypothesis, it doesn't mean it was a waste of time and effort, like I was saying. A lot of times an experiment will bring new scientific questions to think about. And that's how science grows. Okay, it is through the process of trial and error. Something went wrong, so we do it over again with another concept or from another perspective. So scientists usually go through this detailed scientific method over and over again. And as research proceeds, inferences can be drawn and generalizations can be made from the patterns that is observed through all these experiments that you've done. Now, those repeatable experiments, okay, notice that it says repeatable experiments, meaning that you can get the same results multiple times over and over again. Then they will become an impactful scientific results that will be published in peer-reviewed scientific journals to share the knowledge with the public, okay? Scientists are very careful, individuals where we tend to like to, take our results with a grain of salt, because if we claim something to be true and release that particular thing to the public before we confirm it with multiple experiment, it can be very detrimental, lead to a very detrimental consequences. For example, if we think about now in this particular COVID-19 pandemic, people are always thinking about, why does it take so long for the vaccines to come out? Why does it take so long for medications to come out for this particular virus? Well, we can't just say, hey, we're going to put this drug on this one cell and this one group of some, hosts, and then if it works, okay, we're then just going to release it to the public and let everyone take that medication just from that one experiment that works. That is very scary because usually we don't. And that's a whole reason why we have something called clinical trials for medications, is because we need to test it in different stages. Okay, you start with a small group of individuals, then you increase the number of individuals you're testing that drug in to make sure that it not just work for that one small little group or one person. I mean, you have to make sure that it works for everyone, a big group of population of individuals. The other thing you have to think about is side effects. Okay, usually side effects, you can only detect a little bit of them in a small group, of sample size. But if you test that one drug with many people, people, then more side effects will appear. It's like having a bigger sample size so you can better test the results of your drug or medication. So that's a good example of why, experiments has to be repeated over and over again, and why research and discovery of medications takes so long and sometimes years and years is because we have to go through this whole scientific method flowchart here multiple times. And even when we get something that agrees with our original hypothesis, we have to go through it more, maybe 5 more times to make sure that it gives you the same result every single time, that it wasn't just a one-time deal or some kind of, by luck it happened. Okay, so that's why before we release anything to the public in published format, usually it has undergone multiple experiments, okay, through many years sometimes. So these scientific facts that have made it to your textbook today are most likely have gone through many rounds of experiments before someone can claim that it is true. And hence the way that many scientists all over the world are able to use the scientific method to make new discoveries and confirm their finding is what allows us to have all these information we are able to just look up in a book or in a credible educational website. Okay, notice that I mentioned credible, okay, which is something that you should take note of when you write your lab report towards the middle of the semester. Okay, please don't use references that come from .com websites because they are not considered as very credible websites. Take from websites that ends with .edu, .gov, etc. Now, those are more credible educational website that has references there, they cite from these published journals, primary scientific journals, to make sure that what they're saying is true. Okay, not just someone imagining something creatively and then come up with a blog or social media or something and starts claiming something that's absolutely bizarre to the world. Okay, so we have to have credible resources, and credible resources comes from this peer-reviewed journals articles. So what are these peer-reviewed scientific journals that we have just mentioned? Now, these articles usually entails years of collaborative hard work by a group of scientists that have made an impactful scientific discovery for publication. Now, this is different from what we read on the news or social media. Don't confuse primary scientific journals with what's in the news and social media. Yes, even the news sometimes is not completely true facts, okay? I don't know about all the other branches of, business, economics, things like that, but I know my biology. So for science research, I like to go directly to the primary resources in published scientific journals because that is where the experiments were done and the data were collected, just as we have described in scientific method. In addition, a scientific paper before official publication is just a manuscript that must be peer-reviewed before publication. This reviewing of the manuscript is usually done by other scientists that are expert in that particular field of study and is actually a very rigorous and long process, okay, that a manuscript have to go through. For example, in the Journal of Science alone, in this particular case, the statistic is that only 8% of manuscripts are accepted, while 92% of them are rejected. This is how tough it is for a manuscript to make it to publication, which adds to the reason why I like to believe in published scientific journals more than what's on the news when it comes to biology research. Okay, so this reviewing of the manuscript, okay, by other scientists in the field is actually, what we will say that we can really make it highly reliable primary resource. And these right here, what you see, these different squares here are examples of different, more common high-end scientific journals such as the Cell, Molecular Cell, Nature, Journal of Cellular Biology, Cell Biology. These are popular scientific journals that you may see when you visit a clinician's office in the waiting area or something. Okay, so this is why I am more likely to believe in this published journal, like I was saying, more than what they say on the news. Not only was the data from years of research experiments that had to be done multiple times with the same results that we have just discussed, and so also it was read and agreed by other experts in the same field of interest. So what makes up a scientific paper? Okay, so the reason why we go over these different components of a scientific paper right here is because you will have to put the scientific method into practice for lab 5, like I was saying with Daphnia. And then with the data you collect in lab 6 about enzymes, you will have to write your own lab report that includes all these different parts of a scientific paper. But we just introduced this concept in chapter 1. But towards the middle of the semester, feel free to revisit this particular area of chapter 1 in case you forgot what are the components of the scientific paper, which is what you will have to follow in your lab report. Ultimately, these published scientific journal articles are really like lab reports, but just a much more complex level, and usually on things that have not been discovered yet, while the lab reports rewrite in school and education is based on something that's already known and let you have to practice in writing lab reports. So a scientific paper or a lab report that you have to write must include these following components right here. Okay, first, it needs to include an abstract, which is pretty much just a paragraph summary of the entire paper. Okay, I'll further talk about this when I give you an example of a scientific paper. And then the next thing that should be included is what's called an introduction section, which includes background information on your main topic of study. All right, and then the next section is called the materials and methods, which should be detailed experimental techniques that was used. For example, if you use a particular organism to study your research or your project or your lab, or you use a certain instrument to look at certain organism or cells, those are things you include in your materials and methods. And then you have your results section, which includes the data that was collected in your lab. And then the discussion section is what interprets the results. Okay, so results is just the data, the raw data that's collected. Discussion is where you will talk about interpretation of your data. Okay, what does your data mean? What does this bar graph mean? What does this trend in your line graph mean? Okay, so that's what discussion is. And lastly, you have conclusion, which summarizes those important findings from your experiment itself. All right, so now let's try to identify these components in an actual published scientific journal article. So you can apply it to your own lab report. This particular example of scientific paper was from the journal called the Cell. Now, don't be intimidated by the difficulty of this paper. You see all these words. It looks very intimidating, but we're just basically looking at it and identifying the parts. Okay, so don't be intimidated by the information. So before I get to the first part of the component of the paper, I would like to talk about the title. Okay, the title right there. So the title reads Ebola virus epidemiology, transmission, and evolution during 7 months in Sierra Leone. Now notice that this is what we call a very descriptive title. Just from reading the title itself, you kind of have a slight idea what the paper is going to be about. You know about the subject, which is Ebola virus, what they're trying to study about the subject, which is epidemiology, transmission, and evolution of the virus. For how long? For 7 months. And where it was studied? In Sierra Leone. Okay, so when it is your turn to write your lab report in the middle of the semester, one of the criteria I have for your title is a descriptive title, okay? It can't just read Lab 6 enzymes. Well, it doesn't say it. I mean, I only know you're going to test some enzyme of some sort, but what specific type of enzyme are you looking at? Where are you harvesting these enzymes from? What are you studying about the enzyme? So your title, what I mean by a descriptive title, is that it has to be a title where you can identify the subject you're trying to study, a specific subject, and what part of the subject are you trying to find out more about, and how you're doing it. Okay, so that's what you need to practice when it comes to the lab report writing for the descriptive title. Now, an official scientific journal article is usually very long and detailed and, very, like I said, very scary looking with intimidation of all these words, okay? Many pages of words and data. So sometimes a reader just wants to get a quick idea of what the paper is about, because really reading through the whole paper can take hours. So we can't scroll through 50 papers in 50 hours, okay? That takes too long. So that's why every one of these scientific paper has a section called the abstract, okay? So an abstract's purpose is to give the reader a quick idea of what the paper is about, to see if what they're looking for, if this, what they talk about in this paper is what they're looking for. You will have to write your own abstract as well in your lab report. This summarizes your introduction section, experiment section, results, and conclusions in a 3 to 4 sentence paragraph. Okay, so that's why we always like to call the abstract like a mini paper. Meaning that someone that reads your abstract should have a very good idea of what you're going to study, what purpose are you studying the subject for, how you're studying, what was your result, and what was the conclusion, okay, in a quick 3 to 4 sentence paragraph. Okay, and the abstract section in a published journal articles is usually the first paragraph you see on the first page. Now, notice another thing particular about this paper is that almost half the page here, looks scary, is filled with author names and what organizations they're affiliated with. Not a lot of paper looks like this. Usually papers have around 10 authors, but this is a lot. Now, so just from this long list of authors, you know that this research data came from a project that was a really big deal with a lot of collaborative effort from multiple groups of scientists. So that tells you why I say primary research articles are very reliable and credible source of scientific knowledge. Now, after the abstract comes the background or introduction section. And if this was your lab report, you will have to write about some background information on enzymes, what you have learned about the enzymes from lecture or from the lab reading or module itself that you have went through. What specific thing about enzymes are you studying, and what organisms are you getting enzymes from, and what is the name of enzymes you're working with, etc., etc. Okay, I will have specific questions in my instructions for you when you are, about to write your lab report in the middle of the semester. What you need to include in there. I'll be very specific, or else, points will be docked off. A big portion of your grade for lab report will be on the introduction and the discussion section. So this section serves to give the reader a glimpse of your subject of study before they can read further into your experiment. That, gives them a pretty good idea of what enzyme is, for example, and for your lab report, and what reaction you're using to study your enzyme, etc. So the next section on this paper is the data, okay, or results, okay? So now a lot of students get confused about the difference between the data section and the discussion section, even, in their lab reports. The data section is supposed to be really the raw data you have collected during your experiment. So they're usually displayed and compiled into some sort of graphical representation, which is why we have also asked you to graph your results in some sort of a bar graph with data you collect. With the data you collect. You will have first practice with this when you work on your Lab 5 group work assignment. So in addition to post-lab assignments, you have 3 specific group work assignments that you have to share your data with your group members. And then you will divide the job and somebody would have to compile all that data and turn them into graphical representations for your group. Okay, so that will be a good practice for you, and then you will have to use the same concept for the data you collect in Lab 6 for your lab report. And you can see here in these graphs, there's usually, this kind of line, this graph, bar graph, line graph, etc. All of these are considered as raw data that you collect from your experiments, and you're just putting in some kind of illustration format. Then comes the discussion section, which is the part of the lab report that should explain the interpretation of your results or analysis of your data. So again, data section is just the raw data you have collected, while the discussion section is the actual explanation of your data. Not only are you explaining to your readers what your graphs means, okay? Yes, I see that there are color graphs everywhere, but what is the trend? What does the trend tell me? Okay, this is where you will discuss in your discussion section. Also, the other thing you would explain to your readers is if anything was to have gone wrong with your experiment, or if something didn't go as you have predicted. This is also where you will talk about those possible or known errors that have occurred during your experiment. So one example of that in that particular enzyme lab I was talking about is in one of my class section, there was a group that was in charge of looking at effects of enzyme with temperature, and they left the samples out at room temperature for a long time before they put it into the instrument for measurement. Okay, so in that case, that's a source of error. Why their line graphs or reaction rate of their enzyme is not working properly was probably because they did that wrong. Okay, so in that case, those are the type of errors we will talk about in this discussion section. Now, next is the conclusion section, which is usually a very small paragraph right here with a few sentences, just like your abstract, that makes a few final summary ideas of what your discovery was from your experiments. Okay, and this is also another quick glance area for readers to know what their final conclusion is and what the final discovery was at the end of the experiment. So if we go back to our caffeine experiment example, then the conclusion would be that caffeine does increase memory capability, or that caffeine in coffee does stimulate brain activity of some sort. Now, for published journal articles, they like to put the materials and methods section last, but in your lab report, this will be right after your introduction section. And I won't be asking for detailed procedures like how much of something you added to this test tube, how many mils or something like that. I don't expect that. It's just general procedures you did for the experiments, what type of enzymes you used, from what organism, what technique was used to collect and analyze your data. And lastly, the journal article or your lab report will have what's called a reference section, okay? And a reference section, this section is usually for the purpose of information you use in your introduction and background section. Now, when you're talking about those introduction and background information about your topic of study, those ideas or facts did not come from your own creation or imagination. They were somebody else's ideas and facts from somebody else's experiment. So we must give credit to those actual authors for their work, or else you're just taking their information and making them your own, and is considered as an act of plagiarism, which is copying and taking someone else's work as your own without permission. So any information that you have gathered from the internet or the lab module itself, be sure to include a reference for it at the end of your lab report. And in actual published scientific journals, you will always find tons of reference citations at the back here, just like you can see this long section of authors and names of papers, is because there are a lot of things in there that was based off on somebody else's work. And the authors would not chance or want to be accused of plagiarism because, in college as a student, for plagiarism, the worst that can happen is you get a 0 for your lab report. But in a real life situation, plagiarism can lead to, legal matters, okay? Legal matters meaning someone can sue you or you will be laid off your job, okay? So it is very important that we practice and stay away from plagiarism while we are in college, so that when we are in the workforce, we will not practice this at all, okay? All right. All right, so it's been 50 minutes and we have done talking about scientific facts, where do they come from, what is the scientific method, and what is a scientific journal, what are the components, and then introduce what you will have to write in your lab report in the middle of the semester. So let's take a break here for part 1, and then I will start the scientific reasoning in part 2 of this chapter 1 lecture video.